Hello, dear students. I welcome you all on behalf of Sri Jendra Rangjon classes. And dear student, here we are going to talk about today about the fermentation, which is a part of anaerobic respiration. Which is a part of anaerobic respiration. Now it is known that if we talk about a fermentation, then here in fermentation, not much energy released, or too less energy is released. But it is known that fermentation take place in absence of O2, and fermentation is of two type, that is alcoholic fermentation and lactic acid fermentation. These two type mainly we are going to discuss here as according to our syllabus. And this fermentation take place in absence of O2, and that is we can say by the particularly what in absence of O2 during the anaerobic respiration. So fermentation takes place in absence of O2, and two types are there: alcoholic and lactic acid fermentation. Are there. We are going to discuss this individually. First, we are going to discuss the alcoholic fermentation. If you discuss this alcoholic fermentation, then in this fermentation, the incomplete oxidation of glucose take place. In this incomplete oxidation of glucose, incomplete oxidation of glucose is achieved under anaerobic condition. Under anaerobic condition. And in anaerobic condition, this incomplete oxidation is carried out by yeast or done by yeast, that is alcohol fermentation carried out yeast, that is Saccharomyces cerevisiae. Okay, so this yeast carry out this particular thing. And here in this anaerobic condition, a set of reaction take place where pyruvic acid get converted into CO2 and ethanol. Now, how this reaction takes place that we have to discuss. So in the first step, if you talk, then here we know that the glucose is there. Okay, and glucose get converted into pyruvic acid. And as we know, the two molecules of pyruvic acid are getting synthesized. And two molecules get synthesized. Here, during this, as we know, two NADH2 are formed. Two NADH2 are formed. Okay. Now, this pyruvic acid undergo a alcoholic fermentation. Now, what reaction take place that let's understand. So in this, the pyruvic acid, which is there, whose chemical formula and two molecules are there, whose chemical formula is CH3, CO, COOH, that is two molecules are there. Now, in the presence of, we can say pyruvate decarboxylase enzyme, in the presence of pyruvate decarboxylase enzyme, two molecule of CH3CHO and CO2, two molecule of CO2 are obtained. CH3CH2 is a acetaldehyde. Okay, so acetaldehyde get formed, and along with that, two molecule of CO2 are getting released. Then this acetaldehyde get converted into two molecule of C2H5OH, that is ethyl alcohol. And during this, the NADH2, which was synthesized during glycolysis, get used here and converted into NAD plus. And here this process is carried out by an enzyme alcohol dehydrogenase enzyme, which need Zn2 plus to carry out this particular reaction, and two molecule of C2H5OH getting synthesized. Okay, so this is the reaction how proceeds in this. So here the use of two NADH2 molecules, because two acetaldehyde, two molecules of ethyl alcohol get formed, and two molecules of CO2 are released. So in alcoholic fermentation, the release of CO2 is also takes place. Now, in this alcoholic fermentation, it came to know that here the NADH2 which was synthesized in glycolysis get used. So only the two Neet which are obtained in glycolysis, the product is the form during this anaerobic respiration. So in fermentation or specific alcoholic fermentation, not much energy is released. Not much energy is released. Less than 7% of energy in glucose. Less than 7% of energy in glucose is released. And not all of it is trapped, and not all of it is trapped as high energy bonds of ATP. So less than 7% of energy in glucose is released, and not all of it is trapped as high energy bonds of ATP. And it is known that these processes are hazardous. This process is hazardous. This process is hazardous, and yeast poison themselves to death when concentration of alcohol reaches about 13%. Okay, so yeast poison themselves to death when the concentration of alcohol reaches about 13%. So if we inoculate a sugar solution with the yeast carry out alcoholic fermentation, and if concentration reaches to 30%, it can cause a death of the yeast itself. Okay, so this is the way how the alcoholic fermentation takes place. Now we are going to talk about the another type, that is second type, that is lactic acid fermentation. And in lactic acid fermentation, some bacteria, it is carried out by bacteria, and here the lactic acid is going to be produced from the pyruvic acid. So it carried out by bacteria and also take place in muscle cells of animal during exercise. So when oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid by the lactic dehydration enzyme. So if we have a two molecule of pyruvic acid, that is CH3CO COOH, this react with the two NADH2, which is obtained in the glycolysis, and in the presence of lactate dehydrogenase enzyme, it forms two molecules of C3H6O3, that is lactic acid, and along with two NAD are produced. In lactic acid fermentation, it is important that there is a no CO2 release. CO2 release is not observed in this particular, we can say, the alcoholic fermentation. So this is the lactic acid fermentation, and both the two ATP is the net gain of ATP in both the alcoholic and lactic acid fermentation, and that is the ATP which is net gain which is producing glycolysis because energy is going to be used. Now we remember one more important point that during alcoholic fermentation, triose phosphate, that is 3-PGAL, is the electron donor, and acetaldehyde is electron acceptor. Why? Because when 3-PGL breakdown occur, it releases the electron which is used in the synthetic NADH2. That NADH2 again you get used in the what we can say the particular this fermentation. So because of this we can say this is the what a thing during which the acetaldehyde get as the electron acceptor. While during lactic acid fermentation, electron donor is triose phosphate, that is 3-phosphoglyceraldehyde, but acceptor is pyruvic acid. Acceptor is pyruvic acid. Okay, so this way ultimately the particular lactic acid and alcoholic fermentation takes place. So in fermentation, as we have said, yes, the incomplete oxidation of glucose is achieved under aerobic condition by a set of reaction that pyruvic acid is converted to the CO2 and ethanol. The enzyme pyruvic acid decarboxylase alcohol dehydrogenase catalyzed the reaction. As we have discussed, in some bacteria, lactic acid formed by during from pyruvic acid in presence of lactic dehydrogenase. The reducing is NADH2. In both fermentation, merger is not less than 7%, and that we have discussed. When one molecule of glucose is fermented to alcohol or lactic acid, two molecular BT are formed, so net gain is 280. So if we talk about representation, yeast poison themselves to death when the alcohol concentration reaches 13%. When there is a process by which the organism can carry out a complete oxidation of glucose and extract the energy stored to synthesize a large number of ATP molecules needed for the cellular respiration. In eukaryotes, this step takes place within the mitochondria and is required O2. It is aerobic respiration. And aerobic respiration we have discussed already is a process that leads to the complete oxidation of organic substances in the presence of oxygen and release CO2, water, and large amount of energy present in the substrate in the form of 38 or 36 ATP. So this type of respiration is most common in higher organism. That is the thing. Okay, so let's discuss some of the questions. In yeast cells, pyruvic acid is converted into CO2 and methanol by the activity of. So that is the activity of we can say the alcohol dehydrogenase as well as the pyruvate decarboxylase. These both enzymes carry out this reaction. Substrate which undergoes reduction in anaerobic respiration of a yeast, that is alpha ketoglutarate. Okay, acetaldehyde alcohol formation. So substrate which undergo reduction in anaerobic respiration, that is the acetaldehyde. Okay, the formation of alcohol from pyruvic acid is called, obviously, it is the alcoholic fermentation. Yeast poison themselves to the rate concentration of alcohol reaches 13%. When oxygen is inadequate for cellular respiration in muscle, pyruvic acid is reduced to lactic acid by obvious lactic dehydrogenase enzyme. The percentage that is released during the lactic acid and alcohol fermentation present in glucose is less than 7%. Okay, so that is all about the fermentation. I hope you understood that. Thank you, everyone.